Hello, everyone. Today, my group and I will be presenting the third paper of the quarter entitled Hematopoietic Cells Produce BDNF, Brain-Derived Neurotrophic Factor, and Regulate Appetite Upon Migration to the Hypothalamus. A short introduction to our paper. As previously mentioned, BDNF stands for Brain-Derived Neurotrophic Factor, the function of which is to suppress appetite. That is, without brain-derived neurotrophic factor, you or the mice in this experiment specifically go on to develop obesity as a result of lack of appetite suppression, insulin resistance because they're constantly overeating, which is hyperphagia, and chronically high blood glucose level eventually develops into insulin resistance, as we've previously learned this quarter. BDNF is expressed in the parts of the brain that are known to regulate energy balance and appetite suppression, which we'll get more specific as we go on with this presentation, but specifically that's the hypothalamus. Hematopoietic cells that produce BDNF are immature cells that can go on and develop into white or red blood cells, and they are found in the peripheral blood and bone marrow. That's important to keep in mind just as we get into the methods of how this experiment was run and where the experimenters decide to inject BDNF in the mouse models. As previously mentioned, BDNF acts to suppress hunger by acting on the hypothalamic neurons, specifically the paraventricular nucleus of the hypothalamus. Some key concepts as we go into this presentation. The BDNF gene is interesting because it is an example of haploinsufficiency. That is, you need both copies of the BDNF gene in order to effectively express BDNF in cells. So a homo or a heterozygous mutant will not be able to express BDNF. These heterozygous mice, will go on to develop obesity. Humans lacking the BDNF receptor will also develop obesity, and this finding supports the hypothesis that BDNF is directly involved in appetite regulation. We'll get into more specifics as we go on with this presentation, but these are just general concepts, and one of the biggest findings in this paper was that fasting mice expressed higher levels of hematopoietic cells migrating to the paraventricular nucleus, the part of the brain that does regulate appetite and energy balance, energy homeostasis, and they migrate there to express BDNF. This experiment compares the effects of BDNF directly produced by hematopoietic cells as well as BDNF produced by the nuclei in the hypothalamus.
Next is the method section, which will go over the techniques, tools, and statistical analysis. First off, for the animals, the original mice that were used were BDNF LOXP mice, as well as the C57 BL6J mice. These C57 BL6J mice served as the control mice as bone marrow BDNF mice. The knockout mice, which lacked the bone marrow BDNF, were generated and used for the study by the interbreeding of the BDNF LOXP mice to the same background as the control mice. This was done using Cre recombinase of lysosome M to generate the knockout mice. Next was the bone marrow transplant, which used green fluorescent protein, also known as GFP, on the control mice. 8-week-old mice were irradiated and injected with 4 million bone marrow cells. 8 weeks later, the level of positive GFP peripheral white blood cells were counted, and the mice with 60% positive cells were used for the study. Then, 1 month later, the mice were evaluated for positive GFP cells in the paraventricular nucleus, as you can see in figure 3b. Now, the independent variable, which is the fed versus fasted mice. Both groups were taken after a 5-hour fast. Then they were given glucose via intraperitoneal injection. The fasted mice were withdrawn food 16 hours before being euthanized. Both mice were euthanized using the sodium pentobarbital via intraperitoneal injection. Now as for the immunohistochemistry and immunoelectron microscopy. As you can see on the left side are all the chemicals and measurements that were used. Now for the immunohistochemistry, the samples were first perfused with saline to wash out the blood. They were then perfused with fixative in the saline at 4 degrees Celsius. The brain was then immersed 24 hours in post-fixative in saline at 4 degrees Celsius. Next, it was washed with the saline containing sucrose. Then the brain was chopped into 20 micrometer thick frozen blocks and incubated with primary antibodies. It was then incubated with the secondary antibodies that were conjugated to the FITC, CY3, or Texas Red. The next part of this process is nuclear staining, which included DAPI, 2-Pro-3, and propydium iodide. The samples were incubated for 10 minutes on ice. The propydium iodine stained samples were incubated for 30 minutes at 37 degrees Celsius. Now for the immunoelectron microscopy. These were obtained using photomicrographs by using the confocal laser scanning microscope on the 20 micrometer thick slices or a fluorescent microscope. After the post-fixative, the slices were embedded in gelatin. They were then cut into 50 micrometer thick slices on a vibradome for the electron microscopy. The next step is immunoreactivity, which was conducted by reacting tris hydrochloride buffer for 30 minutes at room temperature. Prior to using the electron microscope, the 330 diaminobenzidine stained slices were dehydrated, embedded, and polymerized. They were then stained using uranyl acetate. Finally, the immunoelectron microscopy was conducted. Next is the laser capture microdissection and RNA isolation. RNA analysis was done on 12 micrometer thick frozen sections from either each hypothalamic nucleus or 50 captured GFP positive or negative cells onto a single capture HS laser capture microdissection cap. The RNA was isolated using the PicoPure RNA isolation kit, and the RNA was treated using the DNase-1. For the quantitative RT-PCR, it was executed with the Light Cycler Fast Start DNA Master Green 1 kit and the Light Cycler 480 Pro Master. During the extension phase, fluorescence was measured 3 times for each single reaction. Analysis of the amplification plots were performed using the Light Cycler 480 System 2. For the DNA microarray, it was performed on 5 GFP positive samples, each by laser scanning microscope. From each of the GFP BMT mice, the whole paraventricular nucleus from fed wild-type mice, and the whole paraventricular nucleus from fasted wild-type mice, as you can see shown in figure 2E. Data was analyzed by the gene spring viewer, and the expression ratio R equals fasted over fed was calculated. Finally, for the statistical analyses, the SPS Statistics 19 software was used. On the software, the student's t-test was conducted in order to compare the 2 independent groups. A one-way analysis of variance, repeated measures, analysis of variance, and multiple comparison tests were used to compare 3 or more groups. The statistically significant difference was defined as p-value is less than 0.5.
Okay, so the first thing we're going to look at is figure 1. In figure 1, the scientists explore the idea that fasting induces migration of hematopoietic cells to the PVN after fasting. Okay, looking at figure 1A and B, we see that bone marrow-derived hematopoietic cells, which are microglia-like cells, migrated to the hypothalamus, specifically in the paraventricular nucleus, or the PVN. In figure 1C, we see the comparison of GFP positive cells in mice that were fed normally and fasted. The graph is showing that fasting from 16 hours to 24 hours led to a significantly increased number of GFP positive cells in the PVN. You'll see that the number returned to baseline within 4 hours of refeeding following the fast. This figure right here shows that GFP positive cell density did not change significantly in other hypothalamic nuclei examined during the fast. And then this one is just showing the co-localization of various markers with the GFP positive cells. In figure 1F, it analyzes the percent of marker and GFP positive cells in the PVN in fasted and fed mice. We can conclude from the results that all of these cells were microglial-like, given that glial fibrillary acidic proteins and neuronal markers were not present in the PVN. In figure 2, we see different ratios of expression in the BDNF of fasting mice. In figure 2A, it shows the ratio of BDNF mRNA expression during points of feeding and fasting. The level of BDNF mRNA expression was not changed significantly in other hypothalamic nuclei following fasting. So that's just what this graph is showing here. In figure 2B, an immunohistochemical analysis revealed that the GFP-positive microglial cells also express BDNF. So that's just what the images are showing you here. Immunohistochemistry analysis is a method for demonstrating the presence and location of proteins in tissue sections. In figure 2C, it shows the percentage of the BDNF positive cells located in the PVN and is comparing the absence and presence of IBA, which is a microglial marker. The graph shows that the percentage of the BDNF positive cells found were microglial in nature and found that the increase in BDNF positive cells and fasting was accounted for an increase in the IBA positive subpopulation of BDNF positive cells. So that's why you're seeing a large increase of the BDNF positive cells on the left of the graph compared to the right. Figure 2D shows us the relative expression of different splice variants in the GFP positive cells. Based on the data collected, it was found that type 5 was the major variant produced by the GFP positive cells and that its expression was induced by fasting, which you can also see on the graph. The fasting bar is much higher than the fed bar. Let's see. Figure 2E shows the relative localization of CRH positive neurons and GFP positive cells in the PVN of fasting mice. The small arrows, which are in the center of the screen, indicate the neurons contacted by the dendrites of a single GFP positive cell. The arrowheads, which are on the outer part of the image, show the CRH positive neurons contacted by GFP positive cells. Figure 2F shows the relations of a GFP positive cell and a neuron from the PVN of fasting mice. The small arrow, which is at the bottom of the top image, is pointing to the dendrite of the GFP positive cell and neuron interaction. The larger arrow, which is more to the right, points to the GFP positive cell body. And then the arrowhead at the top left-ish of the screen points to the margin of the neuron. And then the image below that is just showing an illustration of the image they took in this experiment.
Hi everyone, I'm Sarah. I presented the intro to you all, and I am back to present figure 4, which is looking at bone marrow-derived cell-specific knockout of BDNF. So the hematopoietic cells that are derived from bone marrow that produce BDNF are knocked out in these mice. Our first set of figures, we're looking at two graphs separated by gender. The top one is males and the bottom one is females. And the dark dots in these figures, as well as the following figures, represent the knockout mice. That is the mice without BDNF. In these two sets of graphs, the y-axis represents the mice's body weight in grams, and the x-axis represents their age in weeks. These mice were observed from 6 weeks of age to 54 weeks of age, which is just over a year. And throughout this entire time, the knockout mice, the mice without BDNF, had a higher body weight than the mice with BDNF. Again, we're looking at two graphs here. Instead of looking at body weight, we're looking at food intake per day. And the x-axis is the same. We're still looking at age. As you can see, the line with the dark dots representing the knockout mice without BDNF is much higher for both sexes than the line with the white dots, which represents the wild type normal functioning mice. So these two graphs just show that the mice, the knockout mice without BDNF, consumed significantly more food all the way from 6 weeks of age to 1 year of age. Here we're looking at water intake. Again, the mice without BDNF, the knockout mice, consumed more water per day than did their wild type counterparts. Here we're looking at, we'll just start with the two graphs on the left. So again, top and bottom are just separated by sexes, but represent the same variable, and that is blood glucose concentration. The mice without BDNF, represented again by the dark dots, have a higher blood glucose concentration than do their wild type counterparts. For insulin, the next two graphs in the middle, top and bottom again, are just separated by sexes. So top is male, bottom is female. The dark dotted lines are still higher than the white lines, which means that the knockout mice had a higher blood insulin concentration. And that makes sense because these mice without BDNF don't have appetite suppression, similar to what we saw in that study with leptin. The mice without leptin, or in this case without BDNF, are prone to overeating, and that can lead to high blood glucose, high blood insulin levels, and that's why they go on to develop obesity and hyperglycemia. Hyperglycemia, which eventually leads to insulin resistance, as we've learned. The last two bar graphs we're going to look at are of AUC. AUC stands for area under curve, and that just represents how fast a substance reaches the bloodstream. And so, again, the dark bars represent the knockout mice without BDNF, and they had a greater glucose AUC. So more glucose was delivered to their bloodstream faster. That was compared among females and males, but among both sexes, the knockout mice had a greater glucose AUC. For insulin, the bottom right graph, again, the knockout mice had a higher insulin AUC, which means more insulin was being delivered to their bloodstream faster. And the last figure for this section, we are looking at O2 consumption. Interestingly enough, this is the one time the knockout mice did not consume more than their wild type counterparts. Indeed, the knockout mice actually consumed less oxygen both during the day and at night. So, during the day when they're active and at night when they're resting, they consumed less oxygen than mice who were able to produce BDNF.
Figure 5 shows the BMT and intracerebroventricular brain-derived neurotropic factor injections in bone marrow BDNF minus. Figure 5a is the graph to the left, and it shows the therapeutic effect of BMT on BDNF plus BM to BM BDNF minus. Essentially, the body weight decreased in both male and female recipients of BDNF plus bone marrow as compared with those that had received BDNF minus bone marrow. Figure 5b shows that the food intake was significantly decreased in mice that had received BMT from BM BDNF plus mice compared with those that had received BMT from BM BDNF minus. Figure 5c shows the water intake was significantly decreased in mice that had received BMT from BM BDNF plus mice compared with those that had received BMT from BM BDNF minus mice. Figure 5d shows the glucose intolerance improved in mice that had received BDNF plus bone marrow, and figure 5e shows O2 consumption was increased in mice that had received BDNF plus bone marrow. Figure 5f and g show the comparison of average food and water intake about 7 days after the intracerebroventricular injection in male and female mice. And then figure 5h shows the microscopic analysis of the injected GFP positive cells in the PVN, and it reveals that GFP plus bone marrow cells were detectable within the PVN of recipient mice. The left photo shows BDNF plus mice were injected into the third ventricle of BDNF minus mice, whereas the right photo shows isolated MNCs from GFP BDNF minus mice were injected.
The discovery that hematopoietic cell-derived BDNF has a similar effect to neuron-derived BDNF in appetite control is significant to helping people with genetic deficiencies in BDNF production. BDNF reacts on neurons to help regulate appetite and aid in energy balance in the hypothalamus. Hematopoietic cells are also found to migrate to the CNS as a response to different stimuli, which means that hematopoietic cells could be potentially used to solve other illnesses caused by genetic deficiencies. Hematopoietic cells are, in essence, bone-derived stem cells, which can be harvested from other humans or other sources and then given to people with genetic deficiencies. The immediate effects of this particular study could go towards preventing or providing treatment for those with obesity or other eating disorders related to appetite control.
There were 4 main points in the results section of this article. The first one was that fasting induces migration of hematopoietic cells to the paraventricular nucleus. One of the things that made them conclude this was that GFP-positive cells were largely microglia-like. This was indicated by the fact that more than 80% of the GFP-positive cells were positive for IBA1, a central nervous system microglial marker, and Ricinus communis agglutinin 1. The other thing that made them conclude that fasting induces migration of hematopoietic cells was that there was a 170% higher BDNF mRNA following fasting. This was indicated by the fact that splice variant type 5 was the major variant produced by the GFP positive cells, and its expression was markedly induced by fasting. The other thing that indicated to them that there was a 170% higher BDNF RMNA following fasting was that BMMNCs expressed the same variant type 5 that was found in the GFP positive microglial cells. The second result that they had was that BDNF expressing microglia are in contact with paraventricular nucleus neurons. The effect of BDNF producing GFP positive cells was amplified by the contacts that they made with the CRH positive neurons via their dendrites, which led them to that conclusion, to the second result, because numerous CRH positive neurons were in contact with microglia-like cells.
So my results section was the last 2 points, and my first point being that mice with BM-specific, bone marrow-specific BDNF, which stands for brain-derived neurotrophic factor deletion, are hyperphagic and obese. BDNF expression in PVN, paraventricular nucleus, was lowered in BDNF negative mice. Couple other things worth noting is that fasting-induced upregulation did not occur, as well as hematopoietic specific BDNF mRNA is important in appetite regulation during 16-hour fast. My second point is BMT, bone marrow transplantation, rescues metabolic abnormalities of brain-derived neurotrophic factor. Appetite control includes a system of regulatory nodes that branch over the GI system in regions of the brain. This created new insight of pathogenesis of obesity and appetite dysregulation syndromes. Some examples of these syndromes include anorexia nervosa and bulimia.